3 is covering cells and their functions. And this is an overview of the concepts that will be covered in this lecture, including microscopes, the cell structure, membrane transport, protein synthesis, cell division, and concepts related to cancer. The key ideas for chapter 3 will be related to gene codes for proteins. And those are the learning objectives for the current lecture. When we are looking at the cell, we need to define a few concepts. And the first concept that needs to be defined is the fact that the cell represents the basic unit of all life. If we want to look at it like this, we can say that the cell represents the simplest structure that shows all the basic characteristics of life, including the following characteristics as organization and inside metabolism, responsiveness, homeostasis, growth, and reproduction. The science that is involved in studying cells is called cytology. And we started to be able to define the structure of cell and understand the cell function not so far ago, only 350 years ago. In order to be able to understand, observe, investigate, research cells, we need devices that will allow us to examine the structure of the cell, which is, by now you know that is extremely small. We use for that an instrument that is called a microscope. And this instrument has the capability of magnifying structures and allowing us to examine them for those structures that are not really visible to the naked eye. Currently, we have a few types of microscopes available on the market. And I will give you a little bit of insight on each of them. The first one is what is called a compound light microscope, and is the type of microscope that you will see in most commonly used in laboratories. For the compound light microscope, their magnifying capability is up to 1,000 times. Most of the time, they provide that by using 2 lenses, in addition with some visible light that will allow for illumination. And in some cases, those microscopes will have an additional source of light that is an ultraviolet light. Another type of microscope will be the transmission electron microscope, or the TEM, that is using an electron beam in place of the visible light that can magnify an image up to 1,000,000 times. The last type of microscope is what is called a scanning electron microscope, or SEM, that does not magnify as much as TEM, has lower capability, only 100,000 times, and is able to show only surface features. In a way, if you want it, it provides you a 3-dimensional view of an object. So in this image, you can see a comparison image of putting together the 3 types of microscopes. Image A is your typical compound light microscope. Type B will be your transmission electron microscope, and you can see how on top of it we see the cilia, those tiny hair-like structures that over here with the transmission electron microscope you can see how large they are. You can really see their structure. Image C actually is providing you the details of the cilia. And you can see how they actually look as a 3-dimensional structure, as hair-like structures. So let's try and answer the following question. Which microscope is most commonly used in laboratories? Scanning electron microscope, transmission electron microscope, compound light microscope, or the simple light microscope? Correct answer will be option C, the compound light microscope, with the capability of magnifying an object up to 1,000 times. Now that we know how we can study the cell, let's see how actually any cell in our body, regardless of the system or the tissue that that cell belongs to, will be organized. So first component of a cell will be its outer layer, which is called a plasma membrane. Sometimes you will see it, you'll find it in research papers called as cell membrane. This is actually an older term that is not really always distinguishing accurately between the cell's outer membrane and other internal cellular membranes. That's why it was not used anymore and was replaced by the term plasma membrane. So what the plasma membrane is doing is the structure that will enclose the cell content from one point of view, so it restricts the inside environment of the cell, however, is highly and actively participating in many cellular activities. And some of those will be growth, reproduction, and communication between cells. While doing that, actually, the plasma membranes will regulate actively what is allowed to get inside the cell and what is allowed to leave the cell. In some cases, some cells will have specialized systems for uptake or absorption of materials from the outside environment. The outside environment of a cell is called extracellular fluid. In the case of those cells that they have specialized uptake structures, will have tiny projections that are called microvilli. Those projections will increase the membrane surface area, allowing for greater absorption. They are usually found on cells that are involved in that process of active absorption as the cells that are lining the mucosa of the small intestine, and they will promote absorption of digested food into the circulation. Another structure where we will find this type of cells will be the kidney cells, where is an active and continuous process of reabsorption of materials that have been filtered out of the blood. Now let's see what will be some elements, some components of the plasma membrane, because the plasma membrane is not, it's a complex structure on itself. So the main substance of the plasma membrane is a double-layered or bilayered structure formed out of lipid molecules. Those lipids will contain also phosphorus. That's why they are called phospholipids. On the other hand, there are some molecules of cholesterol located between the phospholipids, and those molecules will provide strength and flexibility to the membrane. In the structure of the plasma membrane, we'll have also sugars, carbohydrates, in a small amount, and they are present on the outer surface of the membrane, and they are usually combined with proteins as glycoproteins or with lipids, fats as glycolipids. The function of those carbohydrates is in helping the cell recognizing each other and stick together, attaching to each other. In addition, the plasma membrane has a variety of proteins that will float within the lipid bilayer. Those proteins, some of them will extend over the layers of the membrane, inside and outside the surface of the cell membrane. The proteins on the plasma membrane can be grouped in a few categories. We have channels that are, um, in fact, look like pores in the membrane that allow specific substances to enter and leave. They may be transporters, that they are actioning as shuttle substances from one side of the membrane to the other. They may be receptors. Those are specialized proteins that will mediate the effects of chemical signals on the cell. Chemicals as hormones, neurotransmitters, they will bond in a mechanism of using a mechanism of lock and key to allow for some processes to develop inside the cell. Other will be enzymes that participate in reactions that occur at the plasma membrane level. They may be also linkers. They link to other proteins within the cell, or they may be cell identity markers, because those proteins can be unique for individual cells, and those type of cell identity markers are very important in our immune system. Another component that is essential of the general cell organization will be the nucleus. So the nucleus is the biggest structure, the biggest organelle, the biggest internal structure of a cell. It's surrounded by a membrane that is called a nuclear membrane that encloses its content. And inside the nucleus, every single cell will have a control center that will contain the chromosomes. The chromosomes are thread-like structures that encode the heredity that is passed from parents to offsprings. In addition, the information that is contained in those chromosomes govern all cellular activities. Beside plasma membrane and the nucleus, the cell contains also the cytoplasm. The cytoplasm is a material, gel-like material that fills the inside of the cell. It fills everything between the nuclear membrane and the plasma membrane and will support the remaining organelles. This is a summary again of the structure of the plasma membrane, and you can see in here the bilayer structure with the cholesterol molecules that are spread throughout, giving strength and flexibility, and different types of proteins that function as channels or transporters or receptors. This table summarizes different types of plasma proteins. You have here the channels, the transporters, the receptors, enzymes, linkers, and cell identity markers. Nucleus, beside the structure that I described before of the nucleus, inside the nucleus there is another small structure that is called the nucleolus. And that means nucleolus is, if we need to translate this term, it translates as the little nucleus. The main job of the nucleolus is to participate in assembling ribosomes, which are small bodies that will be released outside the nucleus that are involved in production of proteins. Another slide that will summarize again the cytoplasm structure. The cytoplasm in some cells may present with certain projections. Those can be tiny hair-like structures called cilia. They are short and they move the fluid around the cells, or they can be one long projection that is called flagellum. It has a structure of it looks like a whip extending out from the cell that its main function is to promote the movement of the cell, and it's seen in human beings only in spermatozoa. Let's take a look at the structure of the cell from the point of view of organelles that are suspended in the cytosol. One structure that I already mentioned is the ribosomes. That they are small bodies that are floating free in the cytoplasms, or they can be attached to the endoplasmic reticulum. They are composed of RNA and proteins, and they're involved in manufacturing proteins. Another structure, it will be the endoplasmic reticulum, which can be seen as a network of membranes within the cytosol. It's called rough endoplasmic reticulum when ribosomes are attached to it, or smooth if the ribosomes are not attached to it. And the main function of the endoplasmic reticulum is to modify, fold, and sort protein, while the smooth one that will not be attached to ribosomes is involved in the lipid synthesis. Now we have another structure that is called the Golgi apparatus, or the Golgi complex, which is a large organelle. It looks like a stack of membranous sacs that are stacked on top of each other. The main function of this organelle is to continue the activity of the endoplasmic reticulum and complete the sorting and the folding of the proteins. Mitochondria is the largest organelle that we can find inside the cytosol, and the main function of the mitochondria is to be the energy source, the powerhouse for the cell. We have also lysosomes and peroxisomes that are two different structures, both of them containing enzymes that will digest, will clean, will break down any type of harmful or unnecessary substances that exist inside the cells. Now, different cells will have different structures and different shapes that will actually serve a purpose. The average cell size is about 10 to 15 millimicrons. But also cells may range in size from 7 to red blood cells that are bigger cells that they are 200 millimicrons, or they can be lengthy cells like we can see in the muscle. Definitely one thing we can say about the cells is that the cell shape is related to its function. A neuron, if we need to describe that, let's talk about a neuron or the nerve cell. Has very long fibers that are able to transmit electric signals for very long distances, sometimes up to 1 meter, which is 3 feet. Red blood cells are small and flexible, and they can change their shape, and they are bendable so they can fit inside capillaries that are very small blood vessels. These are a few examples of different types of cells, and you can identify them. A is a neuron that has a long extension, able to pick up and transmit electric impulses. B is epithelial cells that they are flat and they are tied together in a tissue, and their main function is to protect the underlying tissue. We have muscle cells at C that have fibers that are long and strong, and they are able to provide contractions. D are the red blood cells, while E is a sperm cell that is very small, light, and able to swim, to move on its own because it has a flagellum. Let's try and review a little bit. Question number 2 asks, what is the main substance of the plasma membrane? The main substance of the plasma membrane is the phospholipid. Yes, there are some particles of cholesterol, proteins also. However, the main substance is the phospholipid. It's a bilayer. Plasma membrane is a bilayer of phospholipids. Steroids is just a type of hormone. Question number 3. Which organelle generates ATP? The answer is mitochondria. Mitochondria is the powerhouse of the cell. So I was telling you that one of the functions of the plasma membrane is the transportation that can happen across the plasma membrane. Probably the main role of the plasma membrane is to serve as a barrier between the cell and the outside environment. However, nutrients, oxygen, other substances need to get there is a need for those substances to leave the cell or enter the cell periodically, along with, let's say, waste materials from the cell that need to be eliminated. So in this case, we can describe plasma membrane as what is called a semi-permeable membrane because it allows some of the substances to pass where others are kept away. So most substances that are dissolved in intra and extracellular fluid will be soluble in water. If you remember, we discussed in our previous lecture that our body is 90% water. So if so, those substances that can dissolve in water are called hydrophilic. Remember that we said that plasma membrane is made primarily of lipids that are fat soluble or hydrophobic. They reject water. It's just like when you're trying to mix oil and water, they do not mix. So substances that are fat soluble will pass the membrane way easier than those that are hydrophilic. Steroid hormones, gases, those are examples of liposoluble substances. Plasma membrane is a lipid, will allow lipids to go through it. Nutrients, ions, ice, and other hydrophilic substances will not be able to pass through the lipid bilayer alone. They will need what is called a carrier, a transporter, a channel to allow for the cross. We're looking at different types of transport through the plasma membrane. We have two types of those. One will be a passive type of transport, and one will be an active type of transport. What differentiates between those two is the fact that the passive type of transport will not require energy, and it's done towards a gradient from high to low, or from low to high, depending on the concentrations, while the active transportation will be done by expending energy. The cell will need to expend energy for that and can be done even against gradient. In this category, we have the diffusion, which is the net movement of particles from one region of relatively high concentration to one of low concentration. So, in the absence of a barrier, the substances will diffuse down concentration gradients. As you can see, the movement or the flow of a substance will be promoted by a gradient and opposed by its resistance. If you want to think about it, it's just like the filtration systems that you have in your house, or just like the diffusion that happens in a coffee filter. Now, this is a different type of diffusion, and this will need energy for transportation. If you look into this, you can see that we have transporters that will grab some particles, opening on one side, will close, and will carry this particle to the other side. Now, let's talk a little bit about osmosis, which is a special type of diffusion. So when we are talking about this, let's apply it to the diffusion of water through a semi-permeable membrane. Water can move rapidly through the plasma membrane with the help of some channels that are called aquaporins. So it will move, the water will move from an area where there are more of them, more molecules, to an area where there are few molecules of water. So the solvent, the water molecules, will move from an area of lower solute, lower concentration, to an area with a higher solute concentration. From an area with, if you're looking at this, you can see how here I have, if we are taking the example of having two areas with salt water, one with a lower concentration of salt and one with higher concentration of salt. Through this semi-permeable membrane, the water will pass from the higher concentration to the lower, from the lower concentration to the higher concentration until the concentration of particles will equal. So when a solution or an area is defined as isotonic, per definition, it will be an area that has the same concentration of dissolved substances as the fluid in the cell. For example, in our body, um, a normal saline, salty water with a concentration of 0.9% of salt in water or 5% glucose is considered isotonic, has the same tonicity as our inside of the body. Now, when applied, when we are providing the body with this type of solutions, when we are infusing that or we are giving them as an IV, no effect will happen to the cells. And that's because the inside tonicity of the cell and the outside environment has the same level, same concentration of particles, and will keep the cell in an equilibrium with its environment. On the other hand, if we decide to give what is called a hypotonic solution that has a lower concentration of dissolved substances in the same amount of fluid, that is lower than the tonicity, the inside of the cell. And an example of that will be 0.45% saline. The cells will intake the water to equalize the pressures. And as a result of that, the cell will swell and may burst. As an example of that, the red blood cell, let's say, will undergo what is called hemolysis. For a hypertonic solution that has higher concentration of the dissolved substances than the fluid in the cell, that will be a concentration that is over 0.9% salt or over 5% of dextrose. The cell will dehydrate, will lose the water, and will shrink. And the cell will undergo what is called crenation. So if I need to summarize a little bit the concept of osmosis, we are looking at it in terms of water concentration. If I have a low solute, a little bit of salt, and a high amount of water, through the osmosis pressures, the concentration will equalize through a membrane to the second compartment that has high solute and low water by allowing the water to freely pass through the membrane until the solute will reach the same concentration across this membrane. This is the, those pictures will exemplify for you the result of administering isotonic solutions where the concentrations are the same and nothing happened. If I'm giving a hypotonic solution with very low concentration of salt or no salt at all, just water, the cells will swell and burst. While if I'm going a very high concentration of salts and they have a lot of particles dissolved outside, the water will be driven out of the cell and the cell will die through crenation. So now let's look a little bit to the active transport. And the active transport will be a process that requires energy. On the other hand, we can define active transportation as the movement of a solute against their concentration. Normally, they will go from high concentration to small concentration But when we are looking into active transportation, they will move against the gradient from low concentration to high ones. So one example of active transportation that is ATP dependent or energy dependent is the vesicular transportation that is using tiny sacs that are called vesicles. One type of vesicular transportation is endocytosis, when particles that exist in the extracellular fluid will be captured inside the vesicle and transported in the inside of the cell. We have three types of endocytosis. We have a process that is called phagocytosis, where relatively large particles can be engulfed by the plasma membrane, moved inside, and eat inside. Phago means eating. It's typical for the white blood cells in the process of immune response. We have pinocytosis, in which the plasma membrane will engulf small droplets of fluid. Pinocytosis means cell drinking. Or we may have receptor-mediated endocytosis, when the intake of the substances will use specific binding sites or receptors that are called ligands. Another type of vesicular transportation is the opposite way. From the inside of the cell, substances that are stored inside the cells will be eliminated on the outside, and this process is called exo outside, as opposed to the previous one that was called endocytosis. This is called exocytosis, that cells will move materials out with using those vesicles, will move the material outside the cell. So to summarize the processes, we'll have diffusion, osmosis, and filtration that do not require chemical energy. They are what is called passive, and usually the movement of the water will equalize concentration of dissolved particles. And we have the active transportation processes that require chemical energy, require ATP from the cell, that will be the active transportation pumps and the vesicular transportation exo and endocytosis. Let's look at number 4, question number 4. It says chloride enters cells via a channel. What mode of transport is chloride using? Active transport, endocytosis, diffusion, or osmosis? Chloride is using a diffusion type of transportation. Diffusion is a passive type of transportation using a channel. Number 5, question number 5 asks, which method transports small amounts of material into the cell using vesicles? The options are active transport, endocytosis, exocytosis, or osmosis. The correct answer is endocytosis. Endocytosis will transport small amounts of material into the cell using vesicles. Question number 6. Cells chrenate when they are placed in solutions that are catatonic, hypertonic, hypotonic, or isotonic. The answer is cells will chrenate when they are placed in solutions that are hypertonic due to loss of water. To equalize the concentration across the membrane, water would move from inside the cell to outside the cell, while the concentration of particles is increased. Now, another function of the cells is protein synthesis. And we know that proteins are totally necessary as part of the body structure and function. So the cells need to be able to synthesize proteins inside the cell. As I said before, every single one of us has hereditary structures inside the cell that are the chromosomes that are located inside the nucleus. Each of those chromosomes can be divided into multiple units that are called genes. Every single gene carries the message, the information necessary for the development of a particular inherited characteristics. It can be structural, it can be functional, it can be the shape of the eyes, the color of the eyes, the color of the skin, the color of the hair, all those elements. So in other words, in our genes are actually the blueprint for who we are. And our genes will store those characteristics in two substances that are called DNA that can become RNA. DNA is a substance DNA stands for deoxyribonucleic acid. And each one of those particles are made up of subunits. The subunits are called nucleotides. And each one of those nucleotides are made up of 4 basic components. We'll discuss that in a second. Now, there is a related compound that is called ribonucleic acid, or RNA, which is an active participant in the protein synthesis, but it's not part of the chromosome. It's also composed of nucleotides, and the main function of RNA, if I need to describe it, is the fact that RNA will copy a certain sequence of DNA. If the DNA is the blueprint that we have it stored in the nucleus, that blueprint cannot be transferred outside the nucleus. It needs to stay inside and be kept over there for future usage. So the RNA can be seen as a copy of a certain part of that blueprint for a specific protein that needs to be synthesized. Now, this RNA can move outside the nucleus, be transported inside the cell, and inside the cell, it can, based on that information, the cell will synthesize the proteins. So again, we have chromosomes that are made out of genes. The genes are made out of DNA that are made out of this pairing nucleotides that can be adenine with thymine and cytosine with guanine. And each of those nucleotides are made out of a nitrogen base, a sugar unit, and a phosphate unit. So now let's take a quick look and compare and contrast DNA and RNA. In terms of location, if the DNA is almost entirely located in the nucleus, RNA, once synthesized inside the cell, will live synthesized inside the nucleus, will leave the nucleus and will live almost entirely in the cytoplasm. In terms of composition, the nucleotides for DNA will be AGCNT, that stands for adenine, guanine, cytosine, or thymine, with a sugar that is deoxyribose. On the other hand, for RNA, we have the same A, G, and C, adenine, guanine, and cytosine. However, the thymine is replaced by uracil, while the sugar is a ribose, not a deoxyribose. As a structure, the DNA is a double-stranded helix formed by pairing the nucleotide adenine with thymine, and guanine with cytosine, and RNA is a single strand. Now, for function, we have DNA that makes up the chromosomes that are the hereditary units controlling all cellular activities, while the function of the RNA is to manufacturing proteins according to the codes that were copied previously from the DNA. And we have 3 types of major types of RNA. We have the RNA that is M, or messenger. We have the RNA that is R, and the transcription RNA that is T, or translation. So here's an example of the genetic code. And if you look into that, you will see that the glycine, which is an amino acid, the transcribed DNA triplet will be CCC, that will be transferred outside. And you see here different examples of proteins that will have a certain type of DNA code that will transfer outside the body, and the cell will be able to synthesize based on this information the respective protein. We have 3 types, as I was telling you, 3 types of RNA. We have the messenger RNA that is built on a strand of DNA in the nucleus will copy the information from the blueprint, will move outside the cytoplasm, will attach to a ribosome. Now we have inside the ribosome now the protein that is made out by the ribosome will be based on the ribosomal RNA. And we have what is called a transfer or translation RNA that is able to work with other forms of RNA to translate the genetic code into a protein. So in the process of synthesizing a protein, we have two steps. We have the step of transcription, which is the first step, occurs in the nucleus, and the DNA blueprint, the genetic code, will be transcribed into a messenger RNA by nucleotide base pairing. Once this is developed, the translation or the second step will occur outside. The messenger RNA will leave the cell to the outside and in the cytoplasm will connect to the ribosome, and with or without the translation RNA, the messenger RNA will become RNA, rRNA, ribosomal RNA that will be copied and used for production of protein. Again, the DNA is copied by the RNA. RNA will be removed outside and will create the translation and the synthesis of. You can see how it connects to the ribosomes that will start creating the protein chain based on the sequence that they are reading on the um, manufacturing line. It's literally, if you want to imagine something, imagine a manufacturing line, an assembly line. So question number 7, which DNA nucleotide pairs with adenine? And we have for options cytosine, guanine, thymine, and uracil. The answer is the DNA nucleotide that pairs with adenine is thymine. Question number 8, which RNA is transcribed from DNA? Amino acid, messenger RNA, ribosomal RNA, or transfer RNA? The answer is the RNA transcribed from the DNA is the messenger RNA, the one that is inside the nucleus and is able to leave the nucleus and move outside the cell. What is synthesized during translation? Amino acids, proteins, starch, or triglycerides. During translation, proteins are synthesized. The process that I was describing to you is specific for proteins. Now, in the process of growing, repairing, reproduction, the cells of our body will need to multiply, to divide, and increase their number. So for cell division, we have two types of processes. We have one process that is called meiosis that will produce what is called gametes. Those gametes they're very characteristic for our reproductive organs and will cut the number of chromosomes in half. And by doing that, allow for the reproduction because the two participants in the process of reproduction, the gametes need to bring only half of the genetic material, half from the mother and half from the father. So we'll have an offspring containing the right number of chromosomes as a whole body, as a normal human being. On the other hand, we have another process that is called mitosis that is used by what is called somatic cells. All the other cells of our body that are not involved in the process of reproduction are called somatic cells or cells of the body. Somatic means body. And those cells, by reproducing or by dividing themselves through mitosis, will produce cells that have a full luggage of genetic material, will have a 2n number of chromosomes. In other words, the parent cell will divide and will end up producing two identical daughter cells. So in the mitosis, in order to be able to end up with two daughter cells with the same amount of chromosome inside, the cell will need to replicate the DNA and double the amount of DNA. And there are some stages of mitosis. There are 4 stages actually. The first one is called pro, or the beginning, prophase, in which the replicated DNA all the chromosomes will separate from each other, and the nucleolus and the nuclear membrane will begin to disappear. And in the cytoplasm, the the two centrioles will move toward opposite ends of the cell, with a spin-shaped structure starting to develop inside. And that is made out of thin fibers that begin to form between them. So this is the prophase. In the metaphase, meta means in the middle, the chromosomes will line up across the center, equator of the cells, attaching to the spindle fibers, while in anaphase, the centromere will split and the replicated chromosomes will separate and will move toward the opposite ends of the cell. In the last phase, that is called the telophase, a membrane will appear around each group of separated chromosomes, and the 2 new nuclei will be defined. During this stage, also the plasma membrane will start to divide, and in the division, we'll end up from one cells with having 2 cells with the same amount of DNA in each of them. This is the summarizing of it. You see the interphase. When the membrane disappears, the centrioles are moving to the opposite sides, and the chromosomes are putting together in the metaphase. During the anaphase, they will separate and will be pushed and pulled to the opposite sides, while in the telophase, the nuclear membrane starts to form around the genetic material, and the plasma membrane will divide the cells, having at the end 2 cell daughters that have the same amount of DNA as the parent cell. So the question will be, during which phase of mitosis are chromosomes first visible? And the options are anaphase, metaphase, prophase, and telophase. The answer is in prophase. So one concept that will be common among all the systems that we'll discuss during this class and along all the cells in our body is what is called cell aging. So cells are multiplying throughout the life, and some changes will occur that may lead to damage and death of a cell. And those elements that will influence the function of a cell and may harm the cell mainly are known as free radicals that are produced in the course of the normal metabolism. So those free radicals, if they are not counterbalanced by other processes in the body, can be harmful to the cells. Now, as a person ages, our stem cells, our parent cells in our body in different tissues will divide less and less frequently, and the mature body cells will become also less active. And I think that this tiny, cartoon really presents it well, and you can see if the ATP producing organelle, which is the mitochondria, is nice and functional here in a young person. You can see it over here how it's deteriorated and not really able to produce as much as before. And you can see that all the structures become aging. It's just like a machine that becomes old and not functioning as the same level of activity as a machine that is young. Now, there are a few theories related to aging and what makes a cell age. And one of them supports the idea that the cells taken from a young person will divide more times when they are grown in a laboratory environment, as opposed to those um, taken from an older adult. And this can be as a result of what is called programmed cell death, which is named as apoptosis, which is a natural cellular suicide program inside each cell of our body. It's an idea that is trying to say that we have an expiration date from the moment that we are born. So another concept that we'll need to discuss, and we'll approach it every time that we'll describe and we'll discuss a different system, will be the connection between cells and cancer. And among other things, one important element is to understand that regardless what type of cancer we are talking about and where is it in the body, it always starts with one cell that will have a genetic mutation. And due to that genetic mutation, a process that is called hyperplasia will start. There will be an increase in size in the number of cells in that area that is not related to any kind of increased function of that organ. Later, if that hyperplasia is not controlled by the body's immune system, it becomes what is called dysplasia. Now, dysplasia will have cells that actually exhibit already characteristic of cancerous cells, that they have mutations and they have big nucleus and a behavior of a cell that is out of control. If you really look into this, you see that from the beginning and until the stage of invasive cancer, you see that in the picture there is a layer that is not penetrated by the cancer cells, and that is called the basal membrane. And even when we have what is called an in situ cancer, we do see cancerous cells if we do a biopsy in this structure. It's still the basal membrane is not affected. It means that this is what is called in situ, in place, cancer in place. It means that it's not invasive and it's 100% curable. And we like to discover most of the cancers in this stage of being in situ cancers and not when they are invasive. We call an invasive cancer not that much the size of the lesion that we can see or perceive, but by the level of the invasion. The more invasive they are in the other structures around, and if they pass the basal membrane, then this is defined as an invasive cancer. And invasive means that it's a cancer that had the capability of sending out metastasis, which we don't know if they exist or not until we establish and we check the lymph nodes in the region. Now, there will be some elements that can increase the risk for cancer. And those are defined as heredity. Certain types of cancer occur more frequently in some families. The best example is the breast cancer in patients that carry the BRCA gene that increases the risk for breast and ovarian cancer. Some chemicals, and those chemicals that increase the risk for cancers, certain industrial, dietary, or environmental chemicals are called carcinogens. Alcohol is a carcinogen. Smoking, it's another one. Another type of risk factor is radiation and exposure to radiation. Obesity is a risk factor for cancer of the colon and esophagus. Physical inactivity, lack of physical activity will increase your risk for developing cancers, especially of the colon. Poor nutrition is a risk factor for developing more cancers than people that have a normal body weight. And infectious agents, especially viruses, and among them probably the most common one will be the HPV, the human papillomavirus, involved in cancers of both male and female reproductive system. And you can see those summarized in here. Again, heredity, chemicals, radiation, obesity, physical inactivity, poor nutrition, and infectious agents. Question number 11, which of the following is a cancer risk factor? And as an option, we have a diet rich in fruits and vegetables, a family history of cancer, regular exercise, and regular use of sunscreen. The cancer risk factor is a family history of cancer, hereditary aspects of